like to move on and introduce Guy. Guy Cochrane is part of EMBL-EBI, and he is a team leader for the data coordination and archiving group. Guy has been very involved in the COVID data and the upload of SARS-CoV-2 data into the platform. So I think he'll give us some great insights on FAIR data and COVID and some of the changes that may have arisen during the COVID-19 pandemic that we're still obviously discussing and going through. And obviously that might have impact for some of the work that we're doing as well. So Guy, if you're there, I'll hand over to you.
Yeah, so thanks, Serena, and thanks very much, FAIRplus, for the invitation. It's a pleasure to come and present. So I think there's no doubt that the COVID crisis really has raised the awareness and actually the use of scientific data across the board. And so scientific data is known and discussed well beyond scientific communities that would traditionally deal with it. And I think it's almost certainly true that those data sets that get the most exposure and have the most reach are those data sets that can be found easily, are well described, are easy to use, have few restrictions on their reuse, or have clearly defined restrictions on their reuse. And so I think that although many of the consumers of the data may not know the term, and in fact some of the producers of the data may not know the term, actually it is the data sets that have a high degree of FAIR compliance, that are striving for FAIR compliance, that are those data sets that rise to the top and have the reach and impact. So my preoccupation over the last year has been to build, as Serena said, to build the European COVID-19 data platform, which is a foundation to provide data to support our scientific response to COVID-19. And my observations of FAIR, I was asked to talk about the benefits that FAIR might bring in a pandemic scenario, but actually I observe a much more bidirectional relationship between FAIR and the pandemic and our response to the pandemic. So I want to start with three observations. I will describe the platform in a moment, but I want to start with three observations. So the first is that there is no question that we would have been able to move as quickly as we have to mobilize and make available and integrate as many diverse data sets as we've been able to do as quickly and as deeply as we have, without there having been years, decades of previous work from the people around the world whose job it is to organize the data in making those data rich, making those data accessible, describing them and building open services and accessible services on those data. So the fact that there has been FAIRification in the past, the fact that people have strived in the past to meet FAIR principles with their data has really directly benefited our response and our platform. Things have been quicker and deeper because of it. The second observation is that because there's an obvious and clear need for people to integrate their data and to work often across disciplines with data from different disciplines, there's a real use case, there's a real demand. And actually we've seen data sources and those responsible for data sources going through FAIRification processes. So the pandemic itself, certainly as witnessed by our platform, has really driven data sources to become more FAIR, and it's driven people to adopt FAIR principles that might not have considered them before. And then the third observation is that people who have aspirations for FAIR data, who are working in this sphere, who want to bring things together, they come together and work together. So we've seen some fantastically strong collaborations form. And of these three observations, I think the second and third are particularly interesting because these have an impact certainly within COVID, but these will last beyond. Much of the FAIRification, many of the communities will go beyond COVID-19. So when this all ends, there'll be other things that are sustainable that come out of this. So what I'm going to do is introduce the platform. I'll talk about a few of the early priorities, the goals we set ourselves and how we progressed against those. And I'll try to reference back to these observations to give some of the evidence that made me assert these at the beginning. So the European COVID-19 data platform is the European response to provide scientific data in the life science molecular biology domain for the COVID-19 response. It's part of the ERA versus Corona Action Plan. It's one of 10 or so projects in that plan, and it was launched back in April last year. And here, this is the Ursula von der Leyen, the President of the European Commission, launching the platform. And the central premise is really this. It's that if we have data about biological molecules, so biomolecular data, these allow us to identify virus and to understand the movement of the virus, the spread, the transmission of the virus around populations and around the world. And these data also allow us to understand the biology of the virus, the biology of the infection process, and the biology of the human, the patient response to the virus as well. And so with that information, with that knowledge, we can then define better treatment strategies, better interventions, more targeted interventions around public health, and we can design vaccines. And elegantly, because the treatments, the public health interventions, and the vaccine developments, because they tend to use biomolecular assays to measure their effectiveness, then actually we have this elegant return and an elegant virtuous cycle where these cause the production of more data that inform us more, and our knowledge is refined and developed, so we get increasingly good. So we're trying to trigger this cycle to allow data to inform us and better to beat COVID in this way. So the system is multi-layered. For the open biomolecular data, including certainly the data about the virus, these tend to be centralized largely in my institute, the European Bioinformatics Institute so far. So these are centralized data and centralized databases. On the bottom left here, we have national infrastructures. So for biomolecular data that are sensitive, so human patient-connected data, but also for diagnostics and clinical data, these tend to be organized at national infrastructure. The data don't tend to be centralized in any one place. So here we have a set of bilateral connections between the central operation and what's happening in different countries. On the top right here, we have connections into the international, the European level research infrastructures. So molecular biology data are the focus, but of course we need to contextualize this. And so here we have connections through the research infrastructures to, for example, biobanking, to compound screening, to clinical trials, to imaging and so on. So we have strong connections through the European Open Science Cloud into different research infrastructures that provide and organize contextual data. And then at many different levels, there are international global initiatives that connect data and connect services and so on. And then the center of all of this is that we're supporting accessibility of data and tools that help people to access data and use data rapidly as they're produced to produce the most meaningful, to support the most meaningful science to understand COVID and to respond to COVID. So we made a very quick start, and that's because we really didn't start from nothing. We have some strong foundations. And so I'll go through those, and this slide will also serve as this introduces the key partners of the platform. So this is also my acknowledgement slide, I guess. So top left, we have the European Bioinformatics Institute. This is my institute. So for decades, we have been working with international partners to capture and organize and make available the world's biomolecular data, literature about the biomolecular data. Often that involves large data sharing collaborations with America, with Asia, and so on. That's a key partner. Then bottom left, we have Elixir, which effectively provides us with a ready-made network of experts working on bioinformatics data, who are able very rapidly to deploy and work with us to build the system. Top right, we have the European Open Science Cloud, who really provide the connectivity into these different research infrastructures via Elixir, which is our research infrastructure for biomolecular data. So EOS Life is supporting the connectivity with all of this context. And then on the bottom right here, we have a host of different academic, public health, and clinical partners who we've been working with for 8 or so years from my team at EBI to build sequencing, genomics-based pathogen surveillance and pathogen outbreak investigation tools on top of, to layer these specialist pathogen tools on top of the core infrastructure that we provide from EBI. And then finally, the European Commission is very active in operating this project, and the European Union supports us generously through 3 infrastructure projects, Elixir Converge, EOS Life, and the now finished Corbel, and 2 health projects, Recoded and Bio. So there are 3 technical components, or 3 main technical components that we build for the platform. I'm going to talk about the impact of these in the following slides. The first one is the SARS-CoV-2 data hubs. So this is effectively a toolbox for people who are working with viral sequencing data. This allows people to organize, manage their data, to analyze and to visualize and to interpret, and ultimately to share their data into the central system. The central component here, the Federated European Genome and Phenome Archive, is a federated database that allows for those sensitive human data that need to stay within the country of origin. This allows those data sets to be connected and systematically to provide a single accessioning system, searchability across the whole system, and a set of streamlined interfaces for requesting access and managing access request processes, regardless of where the data are actually stored. And the third component on the right is the COVID-19 data portal, and this really serves as the entry point for most people into the system. So this is a website, a set of tools that people use to access the data that are integrated in the system, or to access or to submit their data, to push their data into the system, and to access the various tools. So one of our first priorities was to make data available. So those biomolecular data that will be useful in research in all its diversity against COVID-19. So the first thing to do is to bring data sets that already existed. So perhaps data from other coronaviruses, early data from SARS-CoV-2, host data, and so on, to make all of these available through a single interface in an integrated, navigable way. And so the COVID-19 data portal is the way we do that. It's shown on the right. Here's the link. It's a website. There's a top-level programmatic interface, but actually there is a whole host of different subordinate programmatic interfaces for very highly resolved granular access to different data sources that are connected. And so this is an important combination of tools to provide the right granularity of access. And then we have a growing FTP endpoint, but we're working on other endpoints as well for bulk access to data. And so back in April, we were able already to launch with a lot of data sets connecting to the system, but we've been growing the number of data resources that we capture, and also growing within each of those. There's been a growth of data. Just to give an idea of the span, so we have viral sequences from the virus itself, and sequences from related viruses. We have host sequences from patients and a few from animal models or infected wild animals. We have expression data at the gene level, single cell level, protein level, information about proteins, functional information, structural information. You can just about see on the top right here some protein structure graphics. We have a biochemistry section, so this is about pathways, interactions, and compounds active in those pathways and interactions. Imaging data, and then literature data that relate to molecular biology. And many of these are growing, all of these are growing. Fastest growing is sequence, I'll come on to talk about that. And then we have very rapid growth in the literature domain as well. So this is being used. Here's an example of its use. So this is from the Open Targets initiative. So data available from the portal, such as gene data, target gene data, and compound data, were used by the Open Targets group to run analysis and curation processes to come up with a prioritization tool that lists potential target COVID-19 genes that you might target, and tests that are going on, clinical trials that are going on with different compounds against those targets, ranked by how mature, which stage of trial they're in. in a tool that allows these to be prioritized. So this is a really useful addition for people in therapeutics discovery and development. This has actually come full circle because with working with the Open Targets team, we've been able to put this into the portal as well. So it's now searchable and discoverable also directly within the portal. So another goal from the beginning was specifically about viral data mobilization. Genome sequencing is imperfect, it produces errors. And in order to understand genetic variation reliably, one needs to have systematic approaches to dealing with the error, dealing with noise, excluding noise, and being confident about what's called as a true variation. To do that, one really needs access to raw data. And at the beginning of COVID, there was some sharing of assembled sequences. So assembled sequences are a derived, abstracted form of the data, but very little evidence of sharing and it's making accessible in any way the raw data. So we embarked on a significant campaign, going country to country, to help people who were setting up help people to set up their sequencing operations in different public health labs and hospitals and universities, and help, once they had set up sequencing, to flow their data, to organize data, manage data, and to flow their data into the system. And we did this with the SARS-CoV-2 data hubs. And so we set up 16 national data hubs. You can just about see at the bottom right here in the map. The red boxes are the national data hubs. We focused on Europe first, so there's the bias towards European data coverage. The blue boxes they actually have far smaller numbers in them, but these are data coming from our collaborators. But the red ones clustering around Europe, where we have the greatest density of data, are the data hubs that we've been operating. We've had very rapid uptake of this. It's been very good. This is an example of one of the tools that we provide to help people to push data into the system. But now we have a situation where we have approaching 200,000 sequence libraries, sequence isolates, viral isolates. We have those data available, and the vast majority of the majority of the data that's available to the world. It's come through the system, so we're pleased that this has had an impact. And we now have enough data that we can run some of these systematic analyses. And indeed, yesterday we launched a phylogenetics tool from the data portal that is the first step towards enabling people to visualize that analysis. So this has been used as well. Things are accessible and understandable with a great deal of fair compliance. And the Galaxy project, for example, has picked up the viral data, run analysis workflows to produce variations, they're called variants. This is on the far right, this is from the Center for Genomic Regulation in Spain, the viral beacon. So this is picking up the analysis that was offered by the Galaxy project. It's also running for certain platforms, running its own analysis, and it's providing a navigation tool for people to explore variants and the impacts of those variants in the data set. So this is a really nice example of an ecosystem of services and data processes that are building on top of the portal, using in this case, its programmatic tools, building on top of it and adding some real value and a very nice end user interface here. And so, going back to, just skipping back if I can to these slides. So the fact that, this is an indication that the fact that we could move quickly with this, and already at launch we could show so many different data types, was really a function of the fact that there has been significant effort in the past to make data fair, to make services on the data and to make the data themselves fair. A really good example. But for those data sets and those data sources that we've added more recently, we've been through processes, and the groups that run them have been through processes to enable new tools, to mark up with particular ontologies, to change the approach to presenting data that really represent acts of faiриfication. So this is an example of really where the fact that fair things have happened in the past they've really benefited us and benefited the platform, and where they haven't been happening as required, we've been able to work to improve them. So we've driven verification. And then back to this example, an example of how the portal itself, I think, has presented data in a way that it can attract a great deal of systematic reuse and have a much greater reach and impact. So I think here a demonstration of the fairness of the data. So on to some of these more social aspects, people working together in the national collaborations. So one thing we've begun to build up is a network of data portals. So we have the central COVID-19 data portal on the left here. We now have a network of 5 national data portals. And this is really representing or reflecting the fact that there are activities that are organized and happen at the national level. So for clinical data, for diagnostics data, for epidemiology, but also for funding purposes and for networks of expertise and data management services. There are many things that happen nationally. And so what we do with these national portals is provide, or what the countries that produce them do, is they provide an entry point for scientists within the nation to access those things that they use nationally, the support, the services, the databases, the tools, and so on, but smoothly to connect across to the central COVID-19 data portal for those things that happen more centrally, particularly the open biomolecular data. And so building up this network has been a really interesting and I think fruitful part of what we've been doing. And this is building collaborations that I think will certainly last. A more specific example is the Federated EGA. This is that database, federated database for biomolecular data that are sensitive. We're beginning to see the first nodes emerge, particularly in, so the German, this is the German Human Genome Archive. This is now launched, it's connected. There isn't actually yet any German COVID-19 data in the system. There isn't, the studies have taken the time to start, but it's ready and it's connected. And then here we have the emerging Norwegian effort, and these are the central efforts. So we'll see some more Nordic countries, I think, over the year connecting in their systems. But of course, this is building something that will be useful for COVID-19. But beyond COVID-19, this will be useful in the German case. These are non-COVID-19 studies. But this is already building things that will be useful further into the future beyond COVID-19. So this is a really, really nice sustained impact, I think. This is a pilot from the Netherlands where we are dealing with quite a tricky situation, but there's a great deal of potential. And it's tricky because, so in different parts of the Netherlands, particularly in the university medical centers, they have some very deep and very rich data sets relating to viruses, relating to patients infected with those viruses, lab data, deep sequencing data from host and virus. And so many very deep data sets that are really important and from the same patients. And so there are some really important studies that can be done here where you know the viral variant, you know the genetics of the host, you know about the diagnostics data, you know about the contacts and so on. So some really important things can be done, but these data at the moment tend to be organized very separately in distinct resources. And often the data sets belong to a lab and they're not particularly, there's no real intention to share these data sets. And so what we're doing here in this pilot is using the various tools to provide access to an integrated data set that brings these together, centering on the patient, the albeit still anonymized, but the patient identity. So linking together patient-related data sets and making them available securely in a bit of cloud compute so people can run compute and ask questions across these diverse data sets. So finally, I've mentioned some of the things we're working on, but this is a very big, ambitious project, and there is lots of activity in lots of different directions. It's not possible to talk about all of it in a limited time. Another set of examples of things that we're doing, we are working better to index external data resources and make these more connected into the system. We're already providing cataloging of external resources and linking these together, but we're improving that. We have tools ready. We don't yet have data, but for cohort level studies to support navigation around cohorts. And this is, I guess, a verification process and a data harmonization process for cohorts. Many different tools. We're developing many different analysis tools. There's a lot of curation. This is coming from our Norwegian partners. And then there are data sets for which there isn't an established set of data standards and there isn't established sharing. Here's serology as the example. And we're building out tools that we can use to help to make these data more shareable and more usable and more connectable to the other things we're doing. And then here it's in the middle of a campaign where we are promoting the use of open and fair data resources for these data to make things more usable and more possible in the platform. But I think many or probably all of these will have examples where one is pushing or groups are driven to verify, to observe greater compliance with FAIR principles for their data and for their services and for their tools and for their software, in order to connect things together, to enable, in this case, our platform, but more broadly, to enable the world to use the resources they're putting together. So I think this provides, if you looked at these in detail, they would provide further examples of those objectives, those observations 2 and 3, where FAIR is to a great extent driven by what's happening in terms of our response to the pandemic. So I leave it there, and thank you very much for your attention. I'll be delighted to hear any points or take any questions.
Thank you so much, Guy. Really, really informative talk. Are there any questions for Guy?
I have a question. Hello, Guy. It's Carol.
Hi, Carol.
Hello. Great talk. Really enjoyed it. I think it's one of the best talks I've seen about this, actually. Pulled it all together. I have a question about the analysis. So one of the issues about FAIR was in order to be able to support machine processability and in order to be able to make that easier. Can you give us some sort of points about the ease of being able to pipe data into computational platforms, for example? Did you come across any issues there?
Well, I mean, there are many challenges. So often we're dealing with, particularly with genomics, as you know, we're dealing with large data sets that are difficult to transfer. So we are working on, so to stream data is possible, and people are doing that. But if you want to analyze everything, let's say you want to pick up all of the viral data, all the host data, that's a significant volume of data already. And so you need to engineer systems that will either deal with the bandwidth issues and wait effectively for the data to arrive, or you need to be plugging things in that are more local to the data. And so we have a few examples where we're beginning to use our local cloud. So we're using our local cloud for our compute processes anyway, but beginning to plug in other people's workflows so that some of the compute on the larger data sets can be done locally. In fact, it's important to say that the network of developers for the workflows is very broad. What we have is a workflow engine. People plug their systems into it, and we provide support to do that. But the model really is that the major workflows, where they need to, can operate more centrally. One of the things we're working on is cloud availability, or availability of data, services to make data available more easily in remote compute. And so at the moment, an FTP site is slow, and it's some people need it, but it's slow. We're looking at better ways of having endpoints that can be mounted or made available within different types of compute. And I think a combination of things we do, pre-processing perhaps, and early processing we can do on our side, and making these products available such that they're smaller and more manageable, and then better endpoints. I think that's one of the solutions there. So I think people are succeeding in getting data, but I think we could improve on that.
The endpoints thing sounds really promising, actually. That sounds like a great idea.
Yeah, no, I think it's actually something that for genomics data we really do want to offer. It's too common. The tradition is the FTP, to download data, and it's long been sort of impossible to do that. So one has to do much more filtering, and having fluidity between the databases that we operate for genomics and the kind of compute that people are using is really important. And it's a general problem, so it's one that we can solve generally, so that people don't have to re-engineer it each time.
Hi, Guy. I just comment on that as well. You might have guessed that I might have an opinion on this. So my team at EBI has obviously been supporting Guy with a lot of this work. You know, as some of the people in my team develop some of the back end systems that Guy's mentioned here, and my team is especially involved in this kind of idea of how do we move more of the data into the cloud, but it really comes with a huge opportunity for things like FAIRplus, because in order to do that, you really need to ensure that the data is fair closer to the point of generation. You know, we need to be adopting standards, not, you know, the traditional model of like submit your data to the EBI. EBI will like validate it, maybe curate it up to a standard, and then put it on an FTP site when you get there. Where you do this kind of more cloud-based approach, you push those controls over. Is the data compliant with standards? Is it fair? Much earlier in the process, and I think there's a huge opportunity for the types of processes that FAIRplus has been working towards to assist in that process. And there's the technological component of how do you sort of change the model and adopt more cloud technology in the future, but also this kind of social component about how do you increase the uptake of processes for generating data that's compliant with those standards, and pushing those out broader into the community is something that I think the COVID work that Guy's just talked about is a brilliant example of how to do that, how to sort of increase the adoption of these kind of data hubs. You know, it's a great model, and it's a really nice overlap between the work that Guy's describing here and the sort of process definitions that FAIRplus are sort of trying to work on. So yeah, I think this is only going to get more and more important in future, right?
I think that's a really important point that both Guy and Tony are making, because just making your data fair, here we are, is only part of the activity. It's actually also making it, then the whole point of making it fair was so that it can be processed in a responsible kind of way as well. So so it's not, you know, fair is not the end goal. Fair is just a means to an end. And there's a lot more to do besides, as you were saying there, Tony and Guy.
Yeah, just a quick comment for you, Guy, taking a slightly different approach. So more sort of from the scientific point, I think you've mainly focused more on the viral side and some other biosamples and so forth. From the host genomics perspective, are you connected up with, for example, the COVID-19 host genetics work that Mark Daly and others are doing? Because, you know, I also see it as very much the broad affair, but it's a breakdown of how people are handling their data. So there's a lot more emphasis on breaking down some of the publication barriers and not holding back any of the data that's been produced from an epidemiology point of view for COVID, and some of those more sort of broad changes I think have been brought out by the pandemic, which is great to see that science is changing in that direction. I wonder if you have any broad comments.
Yeah, so we are connected. I think we link at the moment. We have a link out to the registry or the listing that they run, but we're connected really through the European Genome Archive. And I mean, it's true that the viral. So we have prioritized viral data. We felt that was an important thing that was missing. But also, it's true that the human studies take longer to set up, and take longer to yield results. And we're only beginning to see the first. We have a handful of studies, 6 or 7 studies, I think, that have been released for public view. I mean, they're controlled access, but one can look at metadata in the system now. We expect a lot of growth, and we're trying to work with the different projects that in the host genetics network, but elsewhere as well, to include these. And I think over time we will see much more of that. But I think so. I think the fact that you don't see so much of that in the portal yet is really a function of it. it takes longer for those studies to set themselves up. I think when we start to connect into the non-biomolecular data, particularly when we get more deeply into health, we have of course a load of challenges with harmonization and standards and sort of connecting things. We will ultimately be able to, I hope, offer some really nicely connected data sets that span the domains, but I think that, I guess the next step will be host genetics data with a focus, so biomolecular data, and then beyond that it will be the deeper health and the clinical data. They're just naturally more problematic.
Yeah, absolutely. But we're breaking down some of the components, so it's much more rapid assembly of data than we would have seen just a matter of years ago. So it's fantastic progress.